morning, everyone. Okay. So we are here to witness inter-school competition in the name Millet-Utopia, where we have is it fine? Yeah. 800 students in the campus plus 70 faculties from 34 schools. So to inaugurate this event, we are all gathered here, and I request the dignitaries to grace the dais. I request our chief guest, Professor Rajivarsni, Dr. Manoj Prasad, to grace the dais, and I also request our Dean, Professor Sivakumar sir, and our head of the department, Professor Rajagopal sir, to grace the dais. Respected Dean sir, our beloved head Rajagopal sir, distinguished chief guests of today's event, Professor Rajivarsni sir and Manoj Prasad sir, respected faculty colleagues from the University of Hyderabad, and students from different schools in and around Hyderabad, and the teachers accompanying them. It's a matter of great honor for me to welcome you all and express my gratitude for inaugurating this Millet-Utopia, organized as a part of our International Year of Millets celebrations. This program is first of its kind to bring together the school students from class 8, 9 and 10 and raise awareness in them about millets. We sent invitations to approximately 300 schools and received a positive response from 34 schools, which have altogether sent 830 students along with 70 teachers. So in this program we have approximately 830 students plus 70 teachers from 34 schools. As our DST auditorium is under renovation, we could not make everyone to assemble in one place for this inauguration. However, I wholeheartedly welcome the students and teachers to this event. We have two eminent speakers with us today, with whom I am associated with both academically and personally. Professor Rajivarsni sir is a Clarivate Analytics highly cited researcher in the field of agricultural science, specializing in genomics, genetics and molecular breeding. He is engaged in discovering, developing and delivering innovative R&D solutions to tackle the wicked problems faced by global agriculture. Sir has many significant contributions to improving food security in Asia and Africa, and in recognition of his contributions, he has received the highest honors like Shanti Swarup Bhatnagar Prize for Science and Technology by Government of India. Presently, he is International Chair and Director, West Australian State Agricultural Biotech Centre, Murdoch University, Australia. The department feels proud and elated in welcoming you to this event, sir. Today, we also have my PhD supervisor and my lifetime mentor, Dr. Manoj Prasad among us. He is a senior scientist and JC Bose National Fellow at the DBT National Institute of Plant Genome Research, New Delhi. He is also an adjunct faculty in our Department of Plant Sciences, School of Life Sciences, University of Hyderabad. Sir is well known for his contributions to foxtail millet, and in recognition of his contributions, he has received several awards, including the prestigious JC Bose National Fellowship from DST. We are happy to have you here today, sir. At this juncture, I am also pleased to welcome our Dean, sir, Professor N. Sivakumar, and Head of the Department, Professor S. Rajagopal, who are very much supportive and extended their all possible assistances to make this event happen. Last 3 days, I have been running from pole to post to organize this event, and our HOD sir never failed to call me at least once a day to check whether everything is under control or not. He also, I welcome both Dean sir and HOD sir to this event. It would be incomplete if I did not mention the support received from our Honorable Vice Chancellor, Professor Vijay Rao, who readily agreed to my request to organize this event and extended his moral support in this regard. Due to prior commitments, he could not join, but he will be there in the valedictory ceremony. Today, we also have Dr. Mani Vetrivendan among us. He is a senior scientist in the Gene Bank of ICRIS at Hyderabad. He majorly focuses on germplasm conservation and related research, and we will be listening to him post-tea. Welcome, sir. I welcome my faculty colleagues who have joined here with us today. Our MSc and PhD students have made my job easier in organizing this event. A few of them are here to coordinate the event, while the several are working behind the stage. I wholeheartedly welcome them to this event. With this brief note, I conclude my address by welcoming today's chief guests, expert speaker, delegates, faculty colleagues, and participants from different schools. Welcome one and all. Now I request our head of the department, Professor S. Rajagopal, to deliver the opening remarks.
Yeah, good morning, all of you. Distinguished today's chief guest, Dr. Professor Rajiv Varshini, and the invited speaker, Dr. Manoj from NIPGR, and Dean School of Life Sciences, my dear faculty colleagues, and students, and other friends, and university, other staff, and our PR worker. And I welcome you all to the Millet-Utopia. And now this is the entire credit goes to Dr. Muthamilarasan, who has taken initiation to start this program. So just want to say just briefly about this program. So this was about almost 6 months back. We have received a message from the Vice Chancellor through the Ministry of Education that this year was declared as a millet year. So, Vice Chancellor requested us to start a program. Then I thought, I was looking for, the right person. Then I started, luckily, Dr. Muthu is working on, millets. Then we decided that, Muthu will take care of this program. And we call it, this program is a, I can assert not a program. We always say that millet man of university is Dr. Muthu. So we just give him big applause. I think all this credit goes to him. And we have started this program with the, somewhere in October, right? So October started with a big man, a millet man of India. Started with this, and this is the 4th program we are conducting here to celebrate the International Year of Millet. And to say something few about maybe those who are the outsiders, the students and other teachers. So Department of Plant Science, we are about 15 faculties, and 4 are about visiting professors and adjunct professors and other faculties like the honorary professors. And our Dr. Manoj Prasad also is an adjunct professor to the Department of Plant Sciences. And the department is very actively involved in teaching as well as the research. And we would consider among all the universities, we are the topmost department in the country who terrifically publish a very good, very well-known international publications from the university, and recognized DST Feast grant level 2 and the UGC SAP, many other grants. And we are the highest are the fund mobilizers in extramural grants from the the country. So with this, you are all welcome to the Millet-Utopia, and I congratulate once again Muthu for conducting this one, and welcome you all to the Millet-Utopia for the today, and thank you for giving me this opportunity.
Thank you, sir. Now I request our Dean, sir, to spare a few words.
A very good morning to one and all. Distinguished guests on the dais, Professor Rajiv Varshini and Dr. Manoj Prasad, Head of the Department Plant Sciences, faculty colleagues in the School of Life Sciences, and other faculties from other schools and centers, student friends and teachers who have accompanied the students to this place. We are very glad that we are organizing this workshop, a one-day meeting today, particularly the quiz competition for the students, based on the recommendation from the Government of India that this year is declared as the International Year of Millets. And I would like to appreciate the efforts put in by Dr. Muthu Incidentally, his initial also is M. Therefore, M, Millet Man of Plant Sciences, University of Hyderabad. It's very nice that he has been involved right through from the beginning of these programs, and I'm certain that he has drawn a very good schedule for the remaining part of this next year concerning this particular event. And the department also has very rightly selected the distinguished speakers. Both of them are internationally known, and Professor Rajiv was working in an international organization in India, and currently he is also an international scientist in Australia. He came to India, and it's a pleasure to have him here. And Dr. Manoj Prasad also is a very distinguished scientist in the area of plant sciences. He's from the New Delhi Institute. And fortunately, they are also members of our family. Both of them are part of the School of Life Sciences in one form or the other as adjunct professors in the university, and they are not new to the university system, but we are very happy that they were able to make it today. I am certain that you will benefit a lot from their speech talks today morning. As Dean School of Life Sciences, it's my pleasant duty to welcome all of you here this morning. And the School of Life Sciences is one of the largest schools in the University of Hyderabad, with about 67 faculty members and 5 independent departments pursuing different research areas. Each faculty is specialized in a particular area, and we are always in interdisciplinary research activities going on. Though we are for administrative reasons as departments, but it's a unified school, which is, as you see, its entire school is hosting all the 5 departments, and the faculty interact with each other. A lot of students have common courses, and for the school children, it will be very nice to see these facilities around. I welcome all of you to go around when you have some time to look around the school facilities. In other words, we are also a school, but in a different way. We are involved in doing MSc and PhD programs at the University of Hyderabad, and we hope some of you will in future come to University of Hyderabad as students of the University of Hyderabad. All the best for you for the quiz competition, and good luck.
Thank you, sir. Now I request our Dean, sir, to present a memento to Rajiv. Thank you, sir. I request our head of the department to give memento to Manoj Prasad, sir. Thank you. Now, without taking much time, we will move on to the guest lectures. I request the dignitaries to come down. So we will start with Rajiv sir's talk. I request Dr. Pooja Soni, National Postdoctoral Fellow of Department of Plant Sciences, to introduce Dr. Rajiv sir.
Very warm greetings to all the participants and the audience here for this program. I am very excited to see you all here. Thank you to each and every one of you for being here with us today. I feel very privileged to have been given the opportunity by Dr. Muthu to introduce my PhD mentor, Professor Rajiv K. Varshney, on this special occasion. Professor Varshney is an agricultural scientist specializing in genomics and molecular breeding with more than 20 years of service in international agriculture. Currently, he is serving Murdoch University as a director, Center for Crop and Food Innovation Director, State Agricultural Biotechnological Center, and International Chair in Agriculture and Food Security with the Food Future Institute. Prior to joining Murdoch University, Professor Varshney served International Crop Research Institute for Semi-Arid Tropics in India and Africa for 17 years, from 2005 to 22. in various research, management and leadership roles. He has also worked at IPK in Germany from 2001 to 2005 as postdoctoral fellow just after completing his PhD with Professor P.K. Gupta at CCS University, Meerut. Professor Varshney is globally recognized leader for his work on genome sequencing, cataloging, and utilizing genetic diversity, genomics assisted breeding, seed system, and capacity building in developing countries. While working with his colleagues and collaborators at ICRISAT and other organizations around the world, he has made centrally important contributions towards improving food and nutrition security in India and several countries in Africa and Asia by assembling genomes, developing genomic resources, and integrating genomic technologies in crop improvement programs in many tropical crops, and also contributing to deliver several superior crop varieties to some of the world's poorest farmers. Professor Varshney is a highly cited researcher for 9 consecutive years from 2014 to 2022 in a row. He also is an elected fellow to about 10 science and agriculture academic societies in India, Germany, USA, etc. And recipient of several noted awards, including the most coveted science award Shanti Swarup Bhatnagar Prize, the most prestigious agriculture science award. Also, I want to mention one more, Rafi Ahmed Kidwai Award from Government of India, and 2022 International Crop Science Award by Crop Science Society of America. He is the only agricultural plant scientist in India and the only translational plant biologist in Australia to achieve an H-index of more than 100 as per Google Scholar. He has been named as the top scientist in agronomy and crop science category in the list of 250 top scientists in Australia by the Australian newspaper Research Magazine. So thank you very much all for listening. So let us welcome Professor Varshney. Sir, please.
Thank you. Okay, thank you very much. And before I start, let me thank Muthu for his kind invitation to deliver a presentation here. And on this occasion, I would like to thank Professor Shiva Kumar, Dean, School of Life Sciences, Professor Raj Gopal, Head Department of Plant Sciences, and many other senior faculty, including Professor Redenna, Professor R.P. Sharma, also former Head of Department, Professor Venkataramana, Professor Padamja, and several other senior faculty and good collaborators and friends, not only from SLS, from the other disciplines. I'm also very happy to see Manoj Prasad, who has been my old friend from my PhD time, so it's good to have Manoj here. Thank you very much, Pooja, for very nice and elaborative discussion. I'm proud to have a PhD student like you, who is working now with Millet-Man, with Muthu, and also Mani, my another good friend from ICRISAT, so I think this is really pleasure. I'm so happy to see so many students and the teachers from the different schools, and I think these things are required for promoting the awareness and also that about the millets, because from next year we are having the International Year of Millets, but at the same time about the science innovations, so that we can inspire these young students and they can take up their career in science and technology, so that they can benefit society in general, but in country also in particular. So I think, Muthu, you have done a great job. So let's congratulate Muthu and his team for doing wonderful job. And I was talking to Professor Shiv Kumar that Muthu has already organized this memento, so I do not need to give that seminar now. So I can go because I already earned, right? Anyway, so I am working at Murdoch University now, but before that I used to work at ICRISAT, which is an international organization based here in Hyderabad. And some of you may not be aware about that Murdoch University. So very briefly, so Murdoch University is in Western Australia, which is about 40% of the total Australia. We are based in Perth. Here you are having around 5 universities. Our university was established in 73. Next year we are celebrating its 50th anniversary. We are having about 20,000 students with more than 3,000 international students from more than 90 countries. And this is very lively campus. We see lots of young students and they bring a lot of energy. We have the campus in Perth, but also other campuses in Dubai, Singapore and Malaysia. Last time when I was here, I had a meeting with the Honorable Vice Chancellor, also Professor Shiva Kumar and Professor Appa Rao, and we were discussing to develop some MOU so that we can have the exchange of the faculty, students, etc. between these two universities. Now coming back to the agriculture, and I have been working in agriculture for a long time, so we'll discuss some of those importance, role of agriculture, and some of our research contributions, that how we are working in this direction. So for agriculture, at Murdoch University, we have the Food Futures Institute. And in this one, we got 3 different centers: Center for Crop and Food Innovation, which I lead. And in this center, we are having more than 15 different research groups affiliated to this center. And all of them, they are working towards enhancing the crop productivity. Another center is for animal production and health, and this is related to the animal research, animal science, and so. Third is Center for Sustainable Farming System. So these 3 centers, they work together and they contribute towards agriculture, Australian agriculture in that country and at international level. In my center, we are having all these different scientists in the 6 different themes. Each theme is led by one scientist, but in each theme we are having more than one scientist. So we have the different research groups. And these themes, they belong to the crop production and quality, led by Chen Dao Li. Another one is nutrition and food, led by Vicky Solaha. Crop protection and microbiome by Kirsty Bayless. Crop adaptation to climate change and soil constraint by Richard Harper. Digital agriculture by Firdaus Sohail. And impact pathways by Professor Mike Jones. So, and we are having many other scientists working under each theme. So this is our Center for Crop and Food Innovation. I'm leading another center called State Agriculture Biotechnology Center, and this center is a collaborative center for the different university in the Western Australia. And through this center, we provide the state-of-the-art genomics and biotech facilities for different universities, including UWA, Murdoch, Curtin University, and Department of Primary Industry and Regional Development. So this is not just for Murdoch, but the collaborative research center. And we are providing the support to the agriculture and veterinary science. Our university is having a lot of research in the veterinary science. So these are some centers. Now, in terms of my research group, what we are working, so my research group is one of the component of that big center. In my research group now, we are working on wheat. Why? Because all of you who have come here, many of you might have had some nice bread or some paratha or some other thing, and especially for those children, and they should not think that these things come from the supermarket, but they come from the farmer's field. And wheat is very important crop for providing the calories or energy across the world. But in addition to calories, we need to have the proteins, and protein comes from the pulses. So my research group also work on the pulses. And third important crop group is fruit crops, because for minerals, for vitamins, this is another important crop group. So now we work on these fruit crops as well. In terms of the wheat, we are working to improve the wheat grain for the heat tolerance, nutrient use efficiency, drought tolerance, and the frost tolerance, because these are the serious production constraint in the country. So now, if you are having these stresses, the crop productivity will be very low, so we need to improve them. Same thing for the pulses, like chickpea, lentil, fava bean, those crops we are working, and we are working towards improving drought, heat, high protein content, herbicide tolerance. Recently, we have been given a big project from the Hot Innovation Australia. This is about $15 million platform, and this platform called Advanced Genomics Platform for Australian Horticulture. So this is basically for all Australian horticultural crops. But initially in this platform, we will be working on these 5 important crops: banana, pineapple, papaya, custard apple, and passion fruits. So we are interested to enhance the crop productivity, fruit size, etc. Those of you who are working on wheat or pulse crop or on those fruit crops, we are more than happy to collaborate with you in terms of that research. But if you are working on other crops and would like to have the collaboration on advanced genomics, biotech facilities, including GM gene editing, we'll be happy to work with you. What kind of technology we are using? We are working on those genome sequencing analysis. We got the state-of-the-art facilities. We can sequence any genome, not one, thousands of the genome significantly in the less time, in the less cost. Computational genomics approaches are very important to understand the genome. Then we are interested to understand the trait, that why a particular crop is drought tolerant or heat tolerant, what kind of genes are there. So we are working on those aspects. Then we are also working towards improving the crop through the genetic engineering approaches. And another important thing is in the Australia is that linking with the industry, because agriculture in Australia is done by big farmers, large scale holders, unlike India and Africa where agriculture is done by small holder farmers. And then our center is also working for the capacity building for international agriculture. So this is something we are working. But now in the context of this Millet-Utopia function, we would like to share some work which are relevant, more important in international agriculture, and especially from the perspective of India. So now if you think in terms of the food, and I think this is, and how should I say that, for each country this is their obligation to provide enough food to each and every one of us. And India has been doing great work during last several years, producing large amount of food. So food is not a problem now, but distribution, etc. There are other important issues. This is a different thing. But if you think about that, the whole world, then all of us are aware about the issue of the climate change. In climate change, you get very erratic rainfall, drought, heat temperature, and they are not good for the crop production. So we need to develop the crops which can tolerate those climate change impact. Other important thing is that why we need to think about the food. Right now, the world is already having 8 billion population. In next 20 years or so, the number will be reaching about 10 billion population. So the question is that how we are going to feed this world. We need to think from right now. We don't need to do these things 2 years later or 5 years later. So as they say that even right now you see that we already reached now from the 7 to 8, and tomorrow we will be adding more people. So we need to feed these people, and how we are going to do this thing, important thing. Other important point is not only the food, we need to think about the nutrition. So high quality food. Why? Because even if you see right now, then large number of population, about 1 in 3, they are malnourished, and there can be undernutrition, overnutrition, etc. 1.9 billion people, they are having the weight issues, and several million people, they got that issues in terms of the malnutrition. So we need to think this malnutrition from the different perspective. We know the countries like including India, you can see this red color thing, more than 70 million people right now, they're having the type 2 diabetes. Many times when we have the health issues, we go to the physician, we take that medicine. So this is fine. Nowadays you can have the supplementation also in the food. But the thing is, some of these issues, they can also be addressed through the food. If our food is having all different components, then you don't need to be worried about these health issues. So from these perspective, and also this, now there are data that more than this different diseases, dietary risk, they are the #1 cause for maximum deaths around the world. So not HIV, not respiratory infection or cancer, etc. You see on there, dietary risk is the #1. And all these things, they can be taken care by the good quality food. And so diet and nutrition, they play a very important role in this direction. So anyway, in summary, we need to feed 10 billion people by 2050. And not only food, we need to think the nutritious food. And all the different crops, they play important roles. So we do not need to be worried or emphasize only on one crop like rice or maize or chickpea or millets or fruits. We need to have the balanced diet which takes care of all the different ingredients for the nutrition. So we need to always think about the solutions. What are the solutions? Solutions, they are in the hands of large number of people, not only just scientists, also policymakers, farmers, consumers, different things. So we need to see the holistic view that how we are going to do that one. So as I said, if you would like to address the issue of the food, if you would like to address the issue of the nutrition, then probably diet and nutrition, they are the important thing. And unhealthy diets, they are one of the leading causes of the global malnutrition. So we need to think from that perspective that how we can contribute in this direction. Agriculture science community, they have been emphasizing this thing for a long time. And one of the key thing is that we need to think from the diversification of our food crops or so, that in our food, we don't need to be dependent only on 1 or 2 or 3. We need to have this balanced diet. So I think diversification of food is very, very important. And you can see that why we have having all these issues. If you see around the world, or if you see the historical thing, more than 30,000 plants they are available in the world which can be used as a food. But only 7,000 plants they are cultivated for food all over the world. So you are already losing 23,000. But not only that, if you would like to see the commercial scale, only 170 crops they are grown at commercial scale. And only 30 crops they provide the most of the world's calories. So now world is dependent more on these 30 crops, and you would like to go one step further, then only 3 crops, and these wheat, rice, maize, all of them important crops, but they provide 40% of our daily calories. So we need to think that not the ignoring these crops which are important, but we also need to think from the other crops. And nowadays millets, which include sorghum, they also play very important role, and that's the reason. And this was our country India who proposed to have to the United Nations FAO to have the International Year of Millets, and we were also involved from ICRISAT when I was working on that side together with the Ministry of Agriculture, Government of India, that how to move in this direction. When I used to work at ICRISAT, they are working on these different dryland crops, sorghum, pulse millet, finger millet. All these crops, they are very important source of the protein, fibers, minerals. Some pulses crops and legume crops, they are important sources of the proteins. But now in the context of this today's function, Millet-Utopia, we can realize in terms of the importance of those millets, kodo millets, pulse millet, sorghum, and many other things. If you see, and I'm sure that Mani and Manoj, they are also going to emphasize these things, and Mani has done a lot of good work at ICRISAT. You can see that iron and zinc amount, that how they are having very high level of iron and zinc, calcium even 3 times than the milk, same thing in the 2 times from this high in the protein than the milk. So you got a lot of good thing. Now the thing is that earlier time India used to grow a lot of millets, they used to consume, but now this has gone down because of number of reasons. So the question back is that how we can bring them in our food chain again. Other thing is, if you are that thinking from the environmental perspective, then we need to think about the saving the carbon footprints, water footprints. Young generation, they know about the Greta, right? So like these from Nordic countries or so. So we need to see from this perspective that how much carbon footprint and water footprint we can save by using these things. And also from the smallholder farmers, that many farmers, when they don't have the good rain or they don't have the irrigation facilities, then some crops like millets, they can be grown and farmers can get some money from these things. So there are lots of good things. And because of these reasons, as I said, that United Nations, they have announced the 2023 as International Year of Millets. And from Hyderabad University in general, they are doing great work on this direction. Many other organizations, ICRISAT, and of course, Indian Institute of Millet Research, many other people, they are working. So Muthu, you are doing great job. That since you have joined University of Hyderabad, you are working very nicely and promoting millets here. In fact, I think this was last year. So Government of India, Ministry of Agriculture, they are having this magazine called India Perspective. They also asked me to write about the millets, and this magazine goes to the different countries. So basically this is the Ministry of Foreign Affairs, and they were talking, and we mentioned that how these mighty millets, they are the super grains of the power, and they should not be promoted just in India, also in Australia. When I'm working in the Perth, the Council General of India, and we always keep on talking that, well, in the country like Australia, how this promotion of the millets can be done. So I think these things, they play a very, very important role in this direction. Millet is one thing. I have not done much research on millets, only few, limited thing. But Mani and Manoj Prasad, they are going to present. I will like to highlight other area, and this is about the genomics. So for the school children, that genomics basically, what is that one? So how we look, like some of them, that some of you are taller, some of them is shorter in height, some of them, they are having different eye colors, hair colors. So all these things in us, as well as in the different species, this can be animal or plant, they are controlled by the genes, and genes are present in each of you. So basically, if you can understand that entire set of the genes of a particular individual, that's called genome. So we would like to understand the genome of a particular individual, and that's what we say genome. And understanding about the genome and their function, etc., that discipline is called genomics. So genomics is a discipline of the biology, and nowadays all the genes, etc., they are present in the DNA. So you can sequence this DNA through the bioinformatics approaches, computational approaches, you can understand the genes, you can identify the genes. So what we have been doing, that our group earlier when I was at ICRISAT, now in Australia, we have been working on these aspects, that how we can use the genomics technology to understand the genes in the different crops. For that, when I was working here at ICRISAT, we used to work on these three important crops: chickpea, pigeon pea and groundnut, because they are the important crops for the smallholder farmers, but crop productivity was very low. So we always thought how we can enhance the crop productivity. Generally, breeding programs, they make the crosses, and the line which is good, they put in the field, they select this better line, they go to the next generation. But this takes a lot of time. So we always discuss that can we make this process smaller, shorter, and we can do it if you are breeding for drought tolerance, and if you got the genes for drought tolerance in your hand, then you do not need to take every generation in the field. Even in the laboratory, you can screen those plants with the genes and you can tell which line is going to be drought tolerant. So that's in summary. So what we have been doing, we were working on these aspects and we were targeting different approaches. I will like to give some example of these things. So what we did earlier, so you need to understand the genome. So what we did, that we sequenced the genomes of all ICRISAT mandate crops. Either this is chickpea, pigeon pea, pulse millet, and you can see here pulse millet, which is one of the millets, and we wanted to understand the gene. And these are the big process. We are having multi-national consortium, multidisciplinary consortium, but during last few years, we delivered several genomes of all these different crops. And not only these dryland crops of semi-arid tropics, but also several other crops including sesame, mung bean, azuki bean. And this year we had this paper in pea genome also, which is one of the pulse crop. And so we have been working on these aspects. So we have decoded the genomes for more than a dozen crop species. So you can understand, and then by using the computational approach, you can predict that this particular crop is having how many genes. Like in chickpea, we know 28,000 genes. So those are the way that we do it. In the context of millets, pulse millet is one of the important crop. In Hindi, this is called bajra, and bajra is very important in Haryana and Rajasthan and many other parts of that India and also in Africa. So, and bajra is very, very heat tolerant and drought tolerant crop. Many times we grow wheat and rice, and wheat and rice, they belong to the same family of bajra or pulse millet. So by sequencing this pulse millet genome, we also identified the genes for drought and heat tolerance, and we told that how we can use these genes for the different applications. For even you can use these genes to improve heat and drought tolerance in the case of wheat and rice. Recently, we also completed the platinum standard genome assemblies, not one variety which was in 2017, but other varieties also. And you can see now that we can have the genome assembly at the chromosome length. So the whole chromosome, whole genome. So these are the advances in the sequencing technology. So in the case of millets, we contributed something in this direction. The next thing is, if you sequence one individual, is it enough? Or many people think that when we sequence one genome, then you have achieved everything. No, in our opinion, sequencing of one genome is not the end of the story. Rather, this is the start of that game. Why? Just imagine if you sequence this cultivar 1 and you did not sequence cultivar 3 on cultivar 2, then you will be having the genes of only this cultivar 1, some of the genes which are unique to cultivar 2 and cultivar 3. You will not have those genes in your hand. So what happens if you are not taking drought tolerant variety in sequencing program? You will miss the genes for drought tolerance. So with this perspective, we thought we need to sequence more genomes, and when you are having more individuals and when you capture these genes across these different individuals, then the approach called pangenome. We expanded this approach of pangenome to the super pangenome, and here for each species, like in Caesar and chickpea, if you are working Caesar eritinum, this is cultivated, you have the 8 different species, and then you have developed the pangenome of each of these 8 species, and then you put these things together, we call it super pangenome. With this perspective, we started to sequence large number of individuals. So in the case of Pigeon P, we sequenced 300 individuals coming from the 29 different countries. Same thing in the case of chickpea. 430 chickpea lines, they came from more than 60 countries or so. And then we measured or we identified the genes associated with those breeding program related traits like days to maturity, days to flowering, yield under drought and heat stress conditions, etc. So you can understand those genes. Furthermore, we took up another big project, and this was world's largest plant genome sequencing project, where we sequenced 3,366 genomes, and you can see large number of individuals from the different countries, different organizations. So not only that this study made the significant importance in the terms of the science because published in Nature, but more than that, even general media houses, including New York Times, BBC, Economist, etc., and in India many newspapers, they develop the stories that how this kind of significant research or innovation they are going to help agriculture in general and in society in particular also. So these are, and I will not go in the detail, but what I would like to mention that by sequencing all these large number of individuals, you can enhance the gene repertoire. So for instance, earlier we had only 28,000 genes, but now by sequencing additional genome, we developed the pen genomes and we developed additional set of the 1600 genes in this set of those chickpea. Now the next thing is, as I was talking, that we wanted to use, and I used the word translational genomics. So genomics, you can understand the basic function, and I know the contribution from the SLS, many research group leaders, they are making a significant contribution in different areas. So congratulations to all of you. But here the next thing is that how you can use the genomics for the enhancing crop productivity. So how can you translate the genomics for agriculture? So we call this translational genomics. For this perspective, we need to work with the breeding programs. When you talk to the breeders, they say you can screen our population with the genes in the lab, but this will be very expensive. How you can make this process cost effective? So from this perspective, we developed a range of genotyping platforms. So what happens, you put either on the glass slide or on some capillaries, you put those genes and then you screen this large number of population. So we developed this platform called SSR, DART, GBS, whole genome resequencing, etc. So we can do this screening at the DNA level, at the genome level. But the next thing is, you need to have the evaluation of these traits in the field conditions. And we measured large number of these traits in the field. And I'm giving the example of drought tolerance here. So drought is very significant, or this is a serious production constraint. And when I was at ICRISAT, we established different kind of phenotyping platform, and we use these population in the field, in the lysiscan, lysimeter, and also field. I have not put the pictures for other traits, but I just mentioned that Pooja used to work on aflatoxin, and she had very large scale sick plot nursery, and she used to screen the similar kind of population there. At that time, she used to work very hard there in the field. I don't know what is happening now, whether she goes to the field now or not, but maybe in the laboratory, but these are the different contexts. So just I want to mention that at ICRISAT, every student, majority of the people, they used to work in the field and the lab. And Pooja, again, you did great work with us when you were there. So what we used to do, so we have the sequencing data, phenotyping data, and you combine these data together, and then you identify the genes and markers associated with the different traits. So by using these approaches in the chickpea, pigeon pea, and peanut, we identified genes associated with the large number of traits. Now you can see here in chickpea, pigeon pea, or peanut, for different traits, we got the genes in our hands. So we could identify those genes. Nowadays, from this young PhD student, postdocs who are very much interested to advance their knowledge. And I think this is another important thing that nowadays, even when I was at ICRISAT, but now in Murdoch more. So people would like to integrate different omics technology. So either not only the genomics, but proteomics, metabolomics, and nowadays the other approaches, lipidomics, etc. So we need to integrate. And nowadays people can put these things, even artificial intelligence and machine learning approaches. So we need to understand, we need to have the integration of the different technologies. Sometimes we call this approach systems biology as well. So many of us are working on this direction. Here I would like to mention, so not only the QTLs and genes, some of our lab students, and one of the example of one of our former, he completed PhD now, Rutvik. And then what he did, that by using these QTL hotspot for the drought tolerance, because I'm giving the example of only drought tolerance, he put all these approaches together, and we found one of the gene called CATP4V, which is one of the key gene for enhancing drought adaptation in chickpea. So what I wanted to mention, so not only at the QTL level, which are the course thing, but we also did the fine mapping for some of the traits, we reached to the gene level. So you can understand the genetics of the trait. You have the marker platform. Now, we need to think from the farmer's perspective that how you can deliver the better varieties. Farmers, they always need more yield so that they can produce more crop in the field. So from this perspective, we started this approach, and as I said, that genomic breeding approach or translational genomics approaches. In this direction, earlier in genomics assisted breeding approaches, we used to have this concept: marker assisted selection, marker assisted backcrossing, marker assisted recurrent selection. And nowadays we are having new concept of the haplotype based breeding, genomic selection, gene editing for that, promoting the good allele or removing the bad allele. And nowadays you can combine these things together with the speed breeding, so you can enhance several generations in a given years. So we started to use these things, and when we are working, we work with different programs, breeding programs in ICRISAT and national programs in the country, like Indian Agriculture Research Institute in Delhi, Indian Institute of Pulses Research in Kanpur, many other universities, and we started to work with them. And now, for instance, in this particular slide, you can see several chickpea breeders, including Dr. Bhardwaj, Dr. Tripathi, Dr. Soren, and these chickpea breeders, they are coming from the different organization, but then they work together, and we provided our support, and we started to introgress those QTL hotspot for drought tolerance. And while doing these things, these breeding program, they were successful to deliver several superior varieties. I will not go in the detail, but would like to mention that in the case of Ethiopia, for instance, the first successfully, the variety for drought tolerance, Gale 2 was released in 2019. In India, through IRI and ICRISAT, Pusa chickpea 10216. From the US, Raichur Super Nigri 1. Again from that IRI Pusa Manav for the drought and fugitive wilt. In 2020 and 2021, another set of the chickpea varieties, Pusa chickpea 4005, Vijay, IPCL 414, and all these things, they're coming through the genomics assisted breeding. And just few months back, BGM 10221, again from the drought tolerant. And right now, because those of you who are working in agriculture, they have the pipeline All India Coordinated Research Program, ACRIP. Now several of our products, they are already in the pipeline, and just 2-3 weeks back, again Dr. Bhardwaj told me that Pusa JG16, this has also been released. So now, in the case of chickpea alone, more than 10 elite varieties for drought tolerance and fugitive wilt have been released, have been reached to the smallholder farmers, and these are the success stories from the genomics assisted breeding or from the translational genomics perspective. Same thing in the case of pigeon pea. Another variety called Bhima came from the US Raichur by using the molecular breeding approaches. In the case of peanut or groundnut, where oil quality is also very important. So for instance, if you see the peanut oil, then you got the palmitic acid, linoleic acid, oleic acid. Linoleic acid is not good for health. This is for the polysaturated fatty acid. Many American varieties, they got more than 90% oleic acid. So what we did, that by using the genomics assisted breeding, we developed several lines which are having more than 80-90% oleic acid. And India released the first set of those peanut varieties, Girnar 4 and Girnar 5, and they were included in the package of 17 biofortified crop varieties when they were dedicated by Honorable Prime Minister Modi on the 75th anniversary of FAO. So those kind of varieties coming from the genomics assisted breeding, they are being paid attention at the highest level also. So these varieties, they got very less linoleic acid, high level of oleic acid. Same thing for the disease resistance in the case of peanut, and we developed the good varieties for the foliar diseases, and now they are in that ACRIP program. Not only in India, because ICRISAT works in African countries, and we used to have these programs. And when we took several of these lines in the African countries, and before COVID, when I was visiting, and I think this slide is from Ghana, and we could see very nice agronomic performance of these peanut varieties which were developed by us through the molecular breeding and by using these genes, etc. So friends, genomics, by using the genomics approaches, you can enhance your understanding, but at the same time, you can also use these genomics approaches to develop the better varieties, and you can deliver the superior varieties. Now, from where we want to go, and we have given a concept now, and we say this was great, we did a lot of good work, a lot of success stories, but now we need to make this breeding fast forward manner. When we are playing those music nowadays, and if you don't like some songs, you do the fast forward. Same thing, by using these different technologies, you can make the breeding also fast forward manner. How we can do it? Because you can do it because you have a possibility to now to sequence large number of individuals. You have possibility to sequence the populations. And when you are having these sequencing data, you can use, these are the 4 important pillars of the fast forward breeding. From the sequencing to the improved varieties, you can have either haplotype based breeding, genomic prediction, or another important thing is optimal contribution selection, because sometimes when you are taking the parents of the breeding program, they are not very good. If you take good parents, then you don't have good genetic diversity. If you take the good genetic diversity, you don't have the good genetic gain. Now, by using these new approaches of artificial intelligence, machine learning, etc., you can select the parent in such a manner that you are having the good balance. I will talk about these things later. Nowadays, you might have heard a lot about the genome editing, which is very, very powerful approach. They say in coming years, you will see large number of products coming from the gene editing. So what we need to do, we need to understand the genes, we need to understand the basic nucleotide, which is causing the effect on that particular trait, and then you can do the gene editing. India has played a very important role, and now through the DBT, they came up very nice guideline, etc. We missed the boat during the GM thing, but I think this is going to be very good thing for the country now. So you can do that one. And any of these approach which we use, you can combine with the speed breeding. I will like to give the highlight of the haplotype based breeding. So in the case of chickpea, when you had the sequencing data for more than 3,000 individuals, you have a possibility to align those genotypes, and you have the sequencing data for all these genes. You can align, and you can see that for each gene, what kind of haplotypes are present in a given genotype. Now you can see for gene 1, genotype 2 got H3 haplotype, genotype 3 got H4, so like all these different kind of things. Then you can ask a question, which haplotype for a given gene is good? For that, we do the haplopheno analysis, and based on these analysis, we find that okay, for gene 1, though we got 4 haplotypes, but S3 haplotype is good, or S2 haplotype is good, so like that one. Then we ask next question, that the varieties which farmers are growing, whether they got the good haplotypes or bad haplotypes. If some of them bad haplotypes, then we can replace those bad haplotypes with the superior haplotypes. And then we can work, we can improve these elite varieties further by introgressing the superior haplotypes. In this case, as I said, so not only this, we did the sequencing of more than 3,000 lines, we did the phenotyping of more than these 3,000 lines. So what means phenotyping? You put those plants in the field, and then you measure the different traits like yield, seed size, etc. And we did these things at 6 different locations for 2 different years. Then we did understanding of genes, that like which gene is important for the seed weight, which gene is important for the seed yield. Furthermore, by doing this GWAS analysis, we identified these haplotypes for the different genes, like 350 haplotypes for 67 genes. And then we asked that which are those improved varieties, if they got the superior haplotypes or not. But then some of these lines, promising lines from the gene bank, they had the superior haplotype. And when Honorable Prime Minister Modi, when he was at ICRISAT, and he came there to celebrate the 50th anniversary of ICRISAT, but we had a possibility to take the Prime Minister in the chickpea field, and some of those lines, the land races which were having the superior haplotypes, he had that possibility to check them. So I think this is the way now that breeding programs now they can move further to have this introgression of those superior haplotypes in the breeding program. So this is about the haplotype based breeding. The other thing is about the genomic prediction. So earlier we used to predict the performance of a line based on the 1 or 2 gene or QTL. But now you have a possibility to predict the genotype based on the whole genome sequencing, and we have shown that when you do the genomic prediction, you can have the different approaches and you can enhance the trait towards on the right side. So whenever you go, so you can enhance this yield, etc. In the case of chickpea, pigeon pea and groundnut, we also demonstrated from our experiments that how you can enhance the performance, the yield performance at the whole genome level. So this is the way that future is going, and as I said, so now when you got the good gene, you can enhance, you can use that gene editing approaches as well. That's what our center in Murdoch is also working on those aspects. The last thing which I would like to mention, and especially from the developing countries perspective, this is not important that how the variety was delivered, either conventional breeding or through genomics or through the GM approach or through the gene editing approach. Scientist job is many research organization that you develop the varieties. But what happens, these improved varieties, they do not reach to the farmers because of number of reasons. Many of you might have heard about the golden rice. Golden rice are yellow color in that yellow in the color, but they got high level of the provitamin A or so. But because of that policy issues in many countries, this golden rice, though was developed 20 years back, never reached to the farmers, including India. But this year or last year in some countries like in Philippines and Bangladesh, this is moving ahead. What I'm trying to say, we need to make our policies conducive policies so that product is reaching to the farmers in the real time. In India, we have heard the case of the GM mustard. There were lots of struggles. This year, even they got that permission for the environmental release, but then again some problems happened. So I think we need to think from this perspective that how different countries they can address the issue. Second is even the traditional breeding or genomics assisted breeding varieties. Even if you have there are no policy issues or these regulatory issues, etc., but then also you don't have really good seed delivery system. So farmers, they do not get the varieties, and sometimes in some countries they take more than 10 years or so that they reach that good varieties are reaching to them. So we need to develop the good seed delivery system. Even when farmers are given the varieties, but they do not know about the agronomy, that at what time point they need to give irrigation, what time we need to spray the pesticide, etc. Then also they cannot realize the full potential of the genomics. So nowadays with the digital technologies, we can provide the farmers those decision support tools. Nowadays every farmer or all of these different individuals, they got the phones. A lot of people, they are going ahead in providing those different softwares or messages through SMS. Question is, this young generation, they are very much social media savvy. Can they develop the TikTok or Reel or like those kind of things so that these farmers and those people, they can understand that how and what they can do it. So we need to make these things very friendly, and as the country is changing, generation is changing, demography is changing, we need to keep us updating, evolving with the technologies. Same thing that even once you got the agronomy, and if you are having lot of, and in India, in many countries, you got a lot of post-harvest losses, we need to think from this perspective. Also, farmers need to be connected to the market. In many countries and many of these regions, many we have seen that if you got the good rain, then we are having the cost or this price crash, and farmers do not realize the good yield or good price. So we need to think from this perspective. If we would like to provide, we need to provide the good nutrition to consumers, etc. We need to see the value addition, and we need to provide this value addition in the whole chain. So I think not only the rapid discovery system, we need to think rapid delivery system. This is also very important when we are talking from the farmer's perspective, smallholder farmer's perspective. Anyway, in summary, I wanted to mention, as I said earlier, the diversification of crop is very important. Millets play a very important role. Fast forward breeding approaches, they are very important. And nowadays, not only these different approaches of multi-omics or so, we need to think rapid delivery system. We as a scientist cannot do each and everything. That's the reason we need to have the networking, we need to have the partnership, not only with the different research groups or department in the university or institute, but also outside organization, different institute or so, they need to work together. Then we can address these issues. Finally, I would like to thank, because I have been working in this space for last 20 years. During this time, we developed the partnership with large number of partners around the world. So thanks to all of them, all the colleagues. And finally, now in Australia, next year in 2023, we are hosting PAG Australia, so like Plant Animal Genome Conference. Earlier they used to happen only in United States, then they moved to PAG Asia, now PAG Australia. So in the case, if you are interested, please save the dates. Come to Australia or Perth. Perth is beautiful city. Some of you who are working on wheat, then we are hosting 3rd International Wheat Congress in Perth, and we would like to invite you to join us for this meeting. And finally, thank you very much for this, your hearing, and would like to welcome Professor Appa Rao sir also. Thank you very much, sir, for coming here. And so thank you very much once again for all your listening, and we'll be happy to answer any question if you have. Thank you. Yes, madam, please. Can somebody give the mic to? Yeah.
That was an insightful talk, sir. I'm really intrigued to understand why millets were sidelined over the years, why rice and wheat. Could you please throw some light on specific?
Yeah, thank you. So you also asked very good questions. So you asked me to throw some light. I will throw some light. Additional lights will be thrown by Manoj Prasad and Mani, right? So you will also do that one. I think a few things. One is that many people, and these are the sometimes wrong impression of perception, millets have been associated with your societal status. So for instance, that you will be eating bajre ki roti or you will be eating ragi only if you are a poor person or so. And these are coming from those farmers. So this is one. When people, their lifestyle is changing and they have their income, and I think this has happened good in the country, that middle class level has increased. What happens when people get more money, they would like to eat good food, and they think the good food will be made up of the wheat, rice, etc. They go this thing. And not only this one, during last 10 years, even the consumption of the meat, I'm not telling this is wrong, but this has also been enhanced because now people can afford, so they would like to eat that one. Giant food giants, supermarkets, like all these things, they are one thing. Second, even from the farmer's perspective, if they are not getting, earlier these millets, etc., they were grown in those rain-fed conditions. Farmers, they did not have the choice. But now many governments, which is good thing, they say we will provide the irrigation. Many states, they say free electricity, you have the free irrigation. Now farmers have the option to give the irrigation to the field. They think that now they can make more money by growing wheat or rice, why they should grow millets. So I think there are lots of factors. But other thing is, good thing is now that as society is becoming more health conscious, so this millets consumption is started to increase. I think this is very interesting that millets are consumed by very rich group of the people who are going to the supermarket and getting very expensive millets, or very poor people that they are making those roti or so in their villages. And somehow this large number of population which is middle class, they are not taking that one. So we need to target that middle class. Thank you. And additional answer will be given by you guys. No? Okay. Any other question? Yes, madam, please. Oh, this is, yeah. I'm glad that you as a student are asking question. This is very good. From which school you are? Tell your name.
Sir, it was really good.
First you introduce yourself. Your name, your school.
From Vidyanjali High School. I would like to ask a question that the methods you are saying, which is related to genomics, are environment friendly or eco friendly. Would it damage to environment?
So very good question, and I think this is very good question, and all these young students, they should understand that. So as a scientist, okay, what you are saying, this is perfectly fine, and here I would like to classify like development of varieties by three different methods. One is conventional breedings, where you make the cross, you develop the lines. So this is one way. India has been producing all these varieties, etc. They are coming through the conventional breeding. Second is, we were talking about the genomics, where you understand the genes and you use them genes to make your breeding process faster. So here you are not bringing any genes from any outside, etc. So again, there is no regulatory issues here. Third approach is, if you can like to call, that is genetic engineering, which can include both, which is called GM, genetically modified organism. Another approach is gene editing, and both of them, they come in the genetic engineering. In GMOs, some people think, I'm using some people, this can also be the NGOs, that if you bring the gene from other species, put in this particular plant species, they are going to damage environment, they are going to create a lot of problem. Their concern may be right, but as a scientific community, this is our responsibility to address those concerns. For those reasons, each country, they have the regulatory framework, like in India also, and from the Ministry of Environmental and Forest, and there are different committees, and those people can tell. So they take these products and they go very rigorous testing. And this scientific community, which is also led or the scientists, but also the policy makers, when they realize that there is no issue, then only they release. Now many crops in India, especially for the students, from the GM perspective, there is one crop called cotton, BT cotton. In 1960s, 70s, etc., India used to import cotton because we did not have enough clothes. If you see even the old movies, people not wearing these clothes, etc., they were having limited. Even I know that several people in 80s, 90s, people used to have the clothes, new clothes 2 or 3 times in a year. But now you can have every month kind of thing. Why I'm telling, this cotton, India used to import cotton. By using the BT cotton, India has become the net exporter of the cotton. Now India export cotton to the other countries, right? Farmers became beneficiary, country became beneficiary, no health issue like those things. In United States, Canada, Japan, many countries, many food crops, they are also grown by the GM, genetically modified crops, maize, soybean, etc. Crops are grown for more than last 20 years, no health issue, no environmental issues, okay? But some countries like in Europe, there is the issue, some created by NGOs, etc., because of the public, or because of those business interest. In India also, so I'm not saying that we do not need to address the issue, but we need to think, as a scientist, I don't see any problem in the GM crops. Gene editing is another thing. Here you are not bringing any gene from outside, you are doing the editing within the species. Government of India has already came up with some goodish policies, papers or so, and they say that if you are having the gene editing through these methods, we don't have any problem. So you ask this question, you need to read on the internet, but even on the internet also nowadays, those anti-science lobbies or so, they are more proactive. So you will find a lot of wrong information. Internet, this can provide good information, bad information. You as a young student need to see both sides. You need to come up with your own assessment. So now my request to all the young students who are in just 10th, 11th, 12th or so, they need to understand about that one, and you also need to tell this thing to your fellow students and also friends in the other schools. So Muthu, you have done a good job, and I'm very happy that her name was Vidya, or yeah, that she asked the question. So I think we need to have those kind of young minds coming to those scientific conferences.
Any questions from student side?
This time I will give brief answer, so don't worry. Okay, good.
Come little forward.
Again, introduce yourself and then ask the question, please.
I'm Abhita Ghosh of Tatva Global School. I have a question. So in genetic modification, we're changing some genes, we're changing some characteristics. But if we take one characteristic, like one genome, and then add another one, don't these genes like affect each other? Like, let's say that we add something for drought resistance, then can't this affect like something that's about nutrition and then lead to the plant dying quicker? How?
Very good question. Thank you. Thank you for asking this question. I thought I will answer briefly, but again I will have to take some time. But why I'm telling this thing? Because that's so that a student can understand. So in the GM process, you know that which gene you are transferring. Okay? So you are very precise that what kind of changes you are making. When you bring some genes, of course, this can make the impact on the expression of other genes. No doubt about that one. But when you are having very precision integration of the genes, and as I said, you do a lot of testing and you realize that this has not affected this. For instance, in BT cotton when I was talking. So what this BT gene does, that if that insect which was creating the problem for the cotton, and now this, when you bring this BT, then this does not bring this insect come close to the plant. So this is one of the key thing. So you do a lot of testing, etc. And when you are successful to check that this is not a problem, then you go ahead. Now, GM, you have done this thing here because in the laboratory. But if you see the additional varieties or the conventional breeding varieties, the gene transfer from one plant to other plant was happening automatically for many, many years. So this is not the new time. This is happening in that environment historically for many, many years. Whatever varieties, even traditional varieties we see, they are having the gene from one variety in the other one, and you got the composition, you do in the field testing, you do this thing. So to make long story short, rather GM approach is more precise approach for the gene transfer. You know, in the genome where you have transferred, you can also understand its impact. You need to do the different kind of testing, and as I said, even in the India, you have very extensive testing, different committees, and when you see these things that there is no problem, then only you go ahead for the release. Okay? Thank you.
Any more questions? If there are no more questions, then
Now we should give some time to Manoj Prasad also to speak. Manoj is not here, he is gone. Okay, so you can ask more questions till Manoj comes here.
Good morning, sir. I'm Nikhil Yadav from Utopia School, and I want to ask a question, so that, like you have said us many the reasons because of which millets are not even consumed by people commonly, but even millets have many anti-nutrients which make it hard to digest. Could it be a reason that people are not accepting it as in their diet?
I think this is also a very good point. Thank you, Nikhil. So one is that, as I said, so now these food habits, like what kind of food we would like to do, so like quickly, palpatable or taste or digestibility, all these issues are there. So that's the reason in my last slide I was talking about the value addition. So, and nowadays this is happening. So that when we were talking about this bajre ki roti, so you have only this thing that even you make this roti, and you cannot keep this thing for next day or other day because you got the rancidity problem, unlike wheat and rice, etc., that you can save it. Second is, in the villages, or many of us, they know only one way to eat this bajra. Same thing, ragi in Karnataka, we know ragi ball like that one. But nowadays, people need to come up with these new approaches. And nowadays already there, you got ragi dosa, you got different kind of, even paisams, kheer, and all these things, even the cookies, you are having the biscuits. So I think main thing is that you need to come up with these new solution, and nowadays this is new area, agriculture entrepreneurship, and these people, they are coming up with these new ideas that how on one hand we can address the issue of the anti-nutrition. I doubt there are many anti-nutrition things, but the other thing is that what kind of food you can make and how you can have more palpatable, like these kind of thing. So this is the new area, and nowadays the country is doing a lot of work in this direction. Now Manoj is here. Muthu? Yeah. We were waiting for you, Manoj.
thank you, sir. Thank you so much for your
Thank you. It's my pleasure. Thank you very much. Very good.
Good morning, everyone. It is very nice to see many kids here. Hope you are having good time. So indeed, it's an honor to introduce one of my favorite teachers at NIPGR during my PhD, Professor Manoj Prasad. Professor Manoj Prasad was born in Balia, Uttar Pradesh. Currently, he is working as a senior scientist and JC Bose National Fellow at DBT National Institute of Plant Genome Research, New Delhi, India. He completed his graduation, post-graduation, and PhD in botany from University of Calcutta. Prior to start working at NIPGR in 2004, Dr. Prasad served as Alexander von Humboldt Fellow at Leibniz Institute of Plant Genetics and Crop Plant Research, Germany. Dr. Prasad has delivered several invited lectures in different national and international scientific forums and visited various countries like USA, UK, Germany, Australia, Austria, Czech Republic, Italy, Scotland, etc. Dr. Prasad has acquired vast academic and administrative experience as he had held several positions in various academic and research institutions. He is also an adjunct professor in the School of Life Science at the University of Hyderabad. His research interests include plant molecular biology, genetics, genomics, and stress biology. He was one of the pioneers to work on an important underutilized C4 crop, foxtail millet in India. His immense research contributions have transformed this orphan crop into a genomic resource-rich model C4 cereal crop. His work in the area of host pathogen interaction has revealed the molecular mechanism of tolerance in tomato against tomato leaf curl virus. Dr. Prasad has published more than 200 research papers, reviews, and book chapters in highly reputed international journals like Proceedings of the National Academy of Sciences USA, the Plant Journal, Cellular and Molecular Life Sciences, Journal of Experimental Botany, DNA Research, and Theoretical Applied Genetics, Trends in Plant Science, etc. He is also serving as an associate editor in several renowned international journals of high reputation. Dr. Prasad has been recognized with several international and national awards of high repute for his excellent contributions to plant science research. Some of them includes he has received prestigious J.C. Bose National Fellowship, and he is also Fellow of the three national academies INSA, NASI, and NAS. He also received Professor K.K. Nanda Memorial Lecture Award, Birbal Sahni Medal, and Golden Peacock Award, and he also received Tata Innovation Fellowship, National Bioscience Award, Hiralal Chakravarti Memorial Award, and Young Scientist Award from NAS. It is important to note that Dr. Prasad has produced 13 PhD scholars, and several of whom are serving the national institutes and received highest scientific recognition. Although he achieved so much, he is very down-to-earth kind of person, and his office is always open for the students for any kind of help. With this brief introduction, I welcome Professor Manoj Prasad sir to deliver his talk.
So good morning. Still, it is good morning. So good morning to all of you. I would like to thank Muthu for inviting me for this Millet-Utopia, and I also would like to thank my fellow colleague from University of Hyderabad, and other colleagues from other places, and dear students. Actually, that is very important that Muthu has done an excellent job to invite the students from 8 to 10. And I think this kind of things should be done in a very regular basis to grow interest to the kids who are in the younger generation. So today my talk is on fitting the 10 billion by 2050 a genomics perspective. So Rajiv has already set the platform for me, so I will not go in details about it. So yesterday till this time the population of world was 8 billion actually, and it is projected that by 2050 it will be 12 billion. So the question is how to feed the 10 billion because you know that Rajiv has already said that there are several issues like climate change and the recession and this pandemic. So these are the very hard thing. So we have to take care of this thing. So for to survive in this earth, human need these three basic things: either food, shelter, and clothes. Without any of this thing is missing, then it is very hard to survive. And as Rajiv said that food is a distant dream, already told that 821 million are hungry, 1 in 10 among us. And then every 10 seconds a child dies from hunger related issues, and 1 in 4 of the world children suffer from malnutrition. So this is the data of 2019 before the COVID pandemic. So I think the data is still in danger side after this pandemic. So UN has denoted 17 sustainable goal, and the first three, first, second, and third, is directly related to food. So we have to think that how to increase the production and how to save the plants from different natural calamities and diseases. So there are different approach for crop improvement, you can improve the nutritional value, as this Millet-Utopia is there. So we know that all the millets are nutritionally 3 to 5 times superior than the staple crop rice, wheat, and maize. And disease resistance, so improving disease resistance. Like human, we also suffer from influenza and other stuff. Plants also suffer from different diseases. I will show you one example how we decipher the function of one gene. And then the third is improving the abiotic stress tolerance, like heat, drought, salinity. These are all abiotic stress thing. And then improving the agronomic traits, like all the traits like that yield, protein, and these are the things we have to take care of this through improving the agronomic traits. And then last one is improving the water use efficiency and nitrogen use efficiency. If I talk about all these things, millets are having this potential to overcome all these things. So millet are heat tolerant, drought tolerant. They can grow up to 50-degree temperature. So see, now for the last 10 years, the average temperature of India is 30 plus. So millet can be only crop that can grow in this changing climate, in the heat stress. So today I'm going to talk about how we can improve the disease resistance, and the second one part will be how to improve the abiotic stress, and that is the heat stress. So my group works on this crop, tomato, and we work on the plant virus interaction. And in the ongoing research, we have different approach, like small RNA mediated approach for resistance against this tomato leaf curl virus. Another approach is we have used the different gene families like autophagy, dead box helicases, ubiquitin proteasome pathway. And then third one is that epigenetics, RNA dependent RNA polymerase methylation. And then the fourth one is the development of functional marker for developing the gene. So today I'm going to talk about how we decipher the function of one microRNA, which helps the plant protect itself against this deadly virus, tomato leaf curl virus. So this work was done by Navisha Sharma. She is sitting here. So you know that tomato is a very rich source of vitamin A and C and minerals, and also antioxidant like carotene and is lycopene. So this is everyday's meal we used to take. But this plant was suffered heavily by abiotic stresses like this, and then biotic stresses. Among the biotic stresses, the viruses is very devastating. And if we ask about this loss, it can lose 70 to 100%. Once the virus attacks the plant, the whole plant dies. And it was caused by a white fly, transmitted by a white fly, and this is Bemisia tawasi. And the characteristics of this is this, you can see here the curling of leaves in the infected plant. And this is not only for tomato, this is also true from chili and also for papaya. And for the genome organization is concerned, this virus is a single-stranded twin isohedral structure. So this is a DNA virus, single-stranded DNA virus. So there are two kinds of virus, RNA virus and DNA virus. So this is a DNA virus. And this virus has bipartite. It contains two genomes. One is DNA A and DNA B. DNA A is responsible for the virus replication, and DNA B is responsible for the virus movement from one cell to the another cell. So DNA A consists of 6 protein and DNA B is 4 protein. So these are the two varieties we used for our study. This Punjab Chuhara, this is highly susceptible. You can see here the yellow and and this was the farmer's preferred variety because it was size was very similar to brinjal kind of thing, but highly susceptible. And HS8878-1, this is the desi tomato. This is very sweet in taste, but the production yield is not so good like this one. So again, but this is somehow tolerant. But there is no true resistant variety against this virus till date. So to try to understand why one plant in a field is tolerant, another is susceptible. So we check the viral titer after 7, 14 and 21 days of post inoculation in tolerant cultivar and susceptible cultivar. You can see here that 21 days of post inoculation, the tolerant cultivar, the viral titer is less as compared to the susceptible. When we check the synthesis of siRNA, we found that while the tolerant plant, the viral titer is less, but the synthesis of siRNA is more. What does it mean? It means that when the virus attacks the plant, plant synthesizes siRNA against it. If it produces more siRNA against the virus, it wins the battle and becomes tolerant. So in the literature, it was found that there are several TY genes that are providing resistance against this virus, and they has been mapped on 12 different chromosomes of the tomato genome. And these are the 6 genes, TY1 to TY3, this is the allelic, chromosome 6, TY2 and chromosome 11, TY4 in chromosome 3, TY5 and chromosome 4, and TY6 is chromosome 10. So, and they have been mapped here actually. And today I am going to talk about R gene, the resistant gene. It is SW5A, which is not known earlier, and it was located in chromosome 9. No TY genes are there in this chromosome. So first what we did, we checked the presence of these TY genes 1 to 6 in our variety, and we found that this TY1 to TY6 is not present in our variety. Like Punjab Chuhara, it is not present. Even in HS8878, it is not present. So both the tomato cultivar, HS88, the tolerant one and susceptible one, did not carry any of the functional TY gene in a homozygous state. So then we ask the question, what is the thing that why if there is no TY gene, then how this HS8878 is providing tolerance? So we did one small microRNA library, small RNA, and then we identified several microRNA, and this is the expression profiling of this 13 microRNA, and now we have identified one microRNA, that is microRNA 159, which was downregulated in tolerant cultivar and upregulated in susceptible. You can see here 159 is downregulated in tolerant and upregulated in susceptible cultivar. We checked the target of this microRNA 159, and through in silico we identified there are 7 targets, and through RLM race we identified that this microRNA 159 is targeting a transcription factor MYP33. And then we check the expression of this MYP33. As you know that I have told you that this microRNA 159 was downregulated in tolerant and upregulated in susceptible. The target MYP33, since the target in the microRNA is down, the target of MYP33 is upregulated, and this vice versa. And this data was further confirmed by the short tandem target mimic assay, that we have postulated that microRNA 159 possesses the ability to specifically silence the transcription factor MYP33. To check whether this MYP33 is providing tolerance or not, we have used the virus induction gene silencing technique, and we silence this MYP33 in the tolerant cultivar. Tolerant cultivar becomes susceptible. It indirectly shows that yes, it has some role in providing tolerance. This data was further confirmed by this viral titer. When we silence this MYP33 in the tolerant cultivar, you can see here that the viral titer is more as compared to the control plant. And this was also the graph, the same thing. So loss of MYP33 function may assist the viral replication, thereby enhancing the viral infection of the multiplication. So when we silence this MYP33, the transcription factor, the tolerant plant becomes susceptible. Now the question is, what is the function of MYP33 in providing tolerance? Because MYP33 is a transcription factor. So it should have some DNA binding and transcriptional activation. Through YoH-1 hybrid assay, we have shown that MYP33 binds to the MYP binding element, that is TAA-C-AAA. So YoH-1 hybrid assay proved that yes, it has the DNA binding and transcriptional activation assay. To identify the downstream target of this MYP33, we have used the chip sequencing, approach, and we found that there are several MIP binding sites distributed all over the genome. One that 60% of them are in the promoter region, you can see here, and then 34% in the inside the gene and 5% in the transmitter region. When we check the expression of these 21 genes, we found that one gene, that is this Sol9G09813 encoding leucine-rich repeat containing protein, that is known as SW5. The expression is 6-fold upregulated in tolerant and 7-fold downregulated in the susceptible out of all the 21 gene. So we target this gene because since it is highly differentially expressed. SW5 is a coil-coiled NB LRR class of resistant gene. It has 5 paralogs. SW5 confers resistance against orthotoxpovirus. This is a RNA virus in tobacco and tomato. So to check whether this MIP33 mediated regulation of this R gene SW5, we have done electrophoretic mobility shift assay, and you can see here that DNA high affinity DNA protein complex was there when this MIP33 and SW5 is present, and it shows that MIP33 binding on the SW5 promoter may regulate the transcriptional activation of this host R gene SW5. So till this I have shown that microRNA target the MIP33. I have shown through Vicks that when we silence this MIP33 in the tolerant becomes susceptible. In the downstream, that MIP33 binds to the promoter of this host R gene SW5. To check the promoter binding is only for this MIP33, we also use another two R gene, that is TM2 and the N gene, and through this chip sequencing we also found that this SW5 binds to only to the this MIP33, not the TM2, other R gene or N gene. So MIP33 has the capacity to regulate the expression of SW5 in planta. Then we ask the question whether this SW5 play important providing role, and if so, then what is the underlying mechanism for that. Then similarly, like MIP33, we have silenced this SW5 in the tolerant cultivar. Tolerant cultivar becomes susceptible. So it indirectly shows that yes, once we silence this thing, this gene SW5 in the tolerant cultivar becomes susceptible. When we check the viral titer, the SW5 silence tolerant plant, the viral titer is more as compared to the control one. Then we try to find out, to validate this SW5 initiate the hypersensitive response. We actually infect the leaves of the tolerant cultivar with MIP silence, SW5 silenced, and SW5 silenced and MIP overexpressed. And you can see here that no HR hypersensitive response was observed with any of this MIP silenced or SW5 silenced or SW5 silenced and MIP overexpressed. Only in the control condition the HR is seen. So we can postulate that SW5 regulates the hypersensitive response during tomato leaf curl nidillivirus infection. To find out the virulence factor which is responsible for this interaction, we use the 10 viral protein that I have showed you that there are 8 viral protein in DNA A and 2 are in the DNA B. So we use the Yeast-2 hybrid assay to find out the host, which one is the host that viral virulence factor which interact with the host R gene SW5, and we found that AC4 of the viral protein that interact with the host R gene SW5 through this Yeast-2 hybrid assay, and this data was further confirmed by the GST pull down assay and also the co-immunoprecipitation assay. So AC4, that is the viral pathogenesis protein, is the downstream virulence factor of the host R gene SW5. Then we check the viral protein AC4, and we have identified there is a motif through in silico, the RTSK, which is responsible for this interaction with the host R gene SW5, and this was further confirmed by the Yeast-2 hybrid assay. And this further confirmed by the biopsy also, biofluorescence molecular fluorescence assay, that when this SW5 and this AC4 is there, you can see here that interaction is there. When you mutate this viral protein AC4, and the interaction gone. Now the next question is this, that out of these 4 amino acid RTSK, which amino acid is responsible for the interaction? We did site-directed mutagenesis. So when we did the site-directed mutagenesis, we found that serine, which is present in the 48th position, is responsible for this interaction. When we mutate the serine, there is no interaction. Otherwise, when we mutate R, interaction is there, T interaction is there, and K interaction is there. So what is the mechanism for that? That we have shown that in the tolerant cultivar, SW, this microRNA 159 is down-regulated, and that's the region that this target, the transcription factor MIP33 is up-regulated. In the downstream, MIP33 binds to the host R gene SW5, initiate the transcription translation, and the further downstream, this host R gene SW5 interacts with the viral pathogenesis protein, that is the virulence factor, and initiate the hypersensitive response, and it and did the cell death, and that's the reason how it can restrict the viral progression. As contrary to this pathway, microRNA 159 in susceptible cultivar is upregulated. That's the reason its target was downregulated, and the whole downstream process is shut down. So that's the plant become there is no hypersensitive response. That's the plant become susceptible. And this is paper we have published last year in the PNS, and I have shown you that a novel role of microRNA MIP33. So MIP33 is the positive regulator in this case, our case, and microRNA 159 is a negative regulator. Regulate the module that activates the host R gene SW5A, a resistant gene that recognizes the viral virulence factor, that is the AC4 protein, to trigger the hypersensitive response, thereby restricting the virus spread. And 6 months back, she got the scientist position at DBT Institute of Life Sciences. She got recently the Inspire Faculty Out Sir Bowman Excellence Award and INSA Medal for Young Scientist 2019. So we have filed the patent in 2017 also. So another PhD student, Ashish, who works on this autophagy, because autophagy is well worked out in animal system, but very less work has been done in plant system. So recently, for the last 4 years, he has worked out and he has identified several autophagy genes, and autophagy is conserved over the eukaryotic genome. And when he worked out this autophagy, he identified one solar glycopersicum ATG8F, which is also very prominent in animal system. Also, people have several papers on autophagy 8. So he showed that tomato autophagy 8 interacts with the viral protein AC2 trap, transcription activator protein, and degrade this viral thing in the autophagy pathway. So what he has shown that this novel role of the solar glycopersicum ATG8F in providing tolerance against the tomato leukoplakin virus. So what he showed that interaction of this trap, that the viral transcription activator protein with this the host ATG8 leads to its degradation in the vacuole, where it degraded in the autophagy pathway. So this trap and ATG8F, they interact in the nucleus and export this complex into the cytoplasm, and it was phagocyzed by the phagophore, and it matures to the autophagosome, and autophagosome delivers to the vacuole, and it trap was degraded in the vacuole. So these are the basically that reviews based on this work in the virus. Similarly, we have also same thing for this millet genomics also. And one article Ashish and me written about the research and leaders twisted tail. This is very important in the perspective of how a good leader or mentor can give guidance to the students, so that their paths for getting this PhD degree get very smoothly. So my group also worked on the other crop, that functional genomics of the foxtail millet. Here also we work on the 4 different aspects. The functional characteristic of stress responsive gene. As I said that all the millets are very stress resilient crop. So what are the proteins that makes the plant so stress resilient? And then the genetic transformation protocol. For any crop improvement program, you required a good transformation protocol, and we have developed a very good transformation protocol for foxtail millet. You also use the GWAS related trait approach to map these nutritional traits and identify the genes. And then molecular regulation of nitrogen use efficiency. We have a project with Newton Bhabha with University of Cambridge in UK, and we have done a very good work, because in millets there is nothing known about the nitrogen use efficiency in millets. So to do this global analysis of stress responsive gene families in 2012, as Rajiv showed in the legumes, the American and the Chinese, they have sequenced this foxtail millet genome, and that makes our life very easy, so that we can identify certain genes which is response to water, and these are some of the genes, DRAVE, HSP, MIB, Werke, and equivalent. So I will going to talk about the DRAVE and HSP in short. So we have used a pipeline developed by Muthu during his PhD, that quickly to identify the candidate genes, because all the gene families have more than 100 genes. So we cannot characterize 100 genes. We have to find out 1 or 2 candidate genes. And this is the pipeline we used for that, and it will help us to identify the candidate gene. So one of the candidate gene, Charu has characterized, this is DRAVE. So when we, at 2009-10, when this NGS was not there, we have constructed a substratic seed and a library for dehydration stress in foxtail millet, and we identified this transcription factor DRAVE, and which was over the time period, you can see here that the tolerant variety is, the fold detection is more as compared to the susceptible variety. So to identify some functional marker, we have basically did this bulk segregant analysis, and these are the upper 4 are the highly susceptible one, and lower 4 panel are the highly tolerant one, and we identified and 558 base pair position 1 G to A transition to A to G transition of a SNP, synonymous SNP, and we have developed a allele specific marker which can distinguish the tolerant with the sensitive variety. So this is very important in for marker assisted selection. If you identified a marker which is linked with the trait of your interest, you can screen the germplasm at the seedling stage. You don't have to wait till the plant become big. So another recent story, what I thought that I should speak today, but due to this thing I have not spoken. It's a very good story that Roshan has another PhD student. What he has characterized one heat shock protein of foxtail millet. There is 113 heat shock protein in foxtail millet, and he identified one HSP21.9, which is differentially expressed in all the 4 abiotic stress condition, in drought, heat, cold, and salinity. And what he has done, that most important thing is this HSP21 is panicoid C4 specific, actually, not present in rice and wheat. So this is the beauty. If you work in a crop which is stress tolerant, you can identify some transcription factor or genes which is not present in rice and other crops, C3 crops. So what he did, that he deciphered the function of this HSP21, which is a molecular chaperone, and to decipher this, he identified an heat shock factor 2E element which regulate the expression of this HSP21, and this HSP21 is chloroplast localized, and this is the reason why it maintains the chloroplast integrity and protects all the photosynthetic machinery against this heat stress. Once he overexpressed this foxtail HSP21.9 to the rice plant, IR64, not in Pusa basmati, this is IR64, and he showed that only 30% is yield loss as compared to the wild type. So this is a very good candidate, that a gene from C4 climate resilient foxtail millet we can transfer in C3 crops, and the C3 crops become at least at 48 degrees the rice can grow. So he has proposed that the Ceteria italica small heat shock protein 21.9 maintains chloroplast function and improves thermotolerance. So this is the slide I want to show everywhere that I talk about Charu. She got each and everything. So if a student do a good work, he also get the reward and award, and the supervisor get the reward. And Namisha, I have shown you that today her work in PNS last year, so she got a job, and she also got all these awards. Like Muthu is already established himself, so I have not shown all the Foxtel work because Muthu maybe time to time showed his. He also got like Charu all the awards, and that small hits of protein, Roshan, for this work he got the his PhD thesis got the ICR Best Jawaharlal Nehru Award for outstanding doctoral thesis in last year. So once the student do some good work, they can get reward also. And that Ashish has showed that autophagy work, because autophagy was very less worked out in a plant system. So that is the reason I can tell that if something is similar to animal was well worked out, so you can also try in the plant system also. So that he showed that understanding the role of autophagy in conferring tomato-typical nasal tolerance in tomato. So there is that ATGC is both similar to both plant and animal. So I think the genes are highly conserved. The HSPs, autophagy are highly conserved. So one can take the clue from animal system and they can work out in the plant system also. So that's all from my side. Thank you very much. I try to make it very concise and to the point. If there is any question, I'll be very happy.
Any questions from the audience?
They don't want to ask. I have talked about hardcore science. Sir. Yeah. No, this is not transcription. That is the hits of protein, sir. 21.9.
I think I might have missed if you have already said it. Where does it occur more, mesophyll or bundle sheet in C4?
C4, it is chloroplast localized. And it is in the, I think, thylakoid membrane, I think, so most probably.
No, my head said, is it in mesophyll more kind of in bundle sheet, in C4? Both places. That may be interesting.
Yeah. So look, we have.
It implies that there is some change in the what you call the heat sensitivity.
So it is your portrait, sir, because you are a plant physiology person, so you can.
It is known that if some of these C4 plants are exposed to cold temperatures, actually the bundle sheet chloroplasts, they start showing disintegration first.
Okay.
And then it will be followed by So there is already, looks like there is some difference.
Yeah. The original worker is here, Roshan is here. I will ask him to talk to you regarding this pertinent question of the bundle seed at mesophyll, sir. Yeah. So whatever we do, sir, this is the first time we are doing, because no information is there. Nobody works on this crop.